Yeah, so in the next hour, we'll have 4 talks. My name is Malay Basu. I'm an assistant professor in pathology. Um, our first speaker is Alex Rosenberg. He's an associate professor in Department of Microbiology as well as a faculty at Informatics Institute. He's an expert in immune reporter sequencing. And most of you know that he's also very active in bioinformatics education, and he co-directs a course with me. So without further ado, Alex, take it away.
Yes, thank you. Thanks, Malay. Good morning. So today I'm going to talk about work my group is doing to build informatics tools for immunology research. So this is an overview slide. Actually, hang on a sec. There we go. This is an overview slide of the various types or some of the projects that we're working on. These are 4 processes. The first one on the top is bulk repertoire sequencing. This is a form of RNA-seq applied solely to the immunoglobulin gene. So it requires specialized methods for both upstream and downstream processing. We're also doing work on 10x single cell sequencing, combining both gene expression, B-cell receptor, and feature barcoding, where you can understand about the phenotypes of individual cells as well as potentially antigen reactivity. Also, we're working on a cytometric serum bead array, which is a way that we can rapidly profile multiple serum reactivities to different proteins, and this has relevance, especially for COVID responses. And finally, we're also working on a single cell platform, which is a plate-based index sort assay with flow. And we could also, where we determine the phenotype and reactivity of individual cells, but can also sequence the B-cell receptors. And we're working on a pipeline to integrate that into a database called Sphere for sequence phenotype and reactivity. So today I'm going to talk about number 1 and number 3. So I'll begin with, I also want to mention the collaborators with each of these methods that we're doing. So there's many collaborators that I would like to acknowledge here. So I'll begin with B-cell receptor sequencing. Now, there's many applications for this technology. As I mentioned, it's essentially RNA-seq applied to immunoglobulin genes on B cells. And without reading all of these, I can say that this approach is very useful for understanding fundamental lineage questions in B-cell biologies about fate decisions and transforming from one cell type to another. It's also very useful in vaccinology. In fact, we've been using this in both in the flu vaccine, also in the HIV vaccine trial network. And it's also useful for studies in immune system development. One study that we have is evaluating the development of the IgA repertoire in newborns for urban babies versus those from old order Mennonites in a rural farming community. And we're also combining that information with microbiome. So that's a study that's underway currently. So just review about B cells. B cells can express a B cell receptor that is very particular to a particular kind of antigen. They can differentiate into plasma cells, which have the ability to secrete antibodies. And those antibodies can be neutralizing, and they're very specific to a certain protein. And the variable part of the antigen, or the B cell receptor, is shown here. And it is the combination of 3 gene elements that are put together during B cell development. And the diversity of the B cell receptor or the antibody is based on multiple possible V genes, D genes, and J genes being combined randomly during development. So that's the first level of diversity. During an immune response, naive and inexperienced B cells that with T cell help and antigen presentation can proliferate and engage in somatic hypermutation to modify these receptors, and those with higher affinity are selected for, and then can further differentiate into either a memory B cell or a plasma cell, which can secrete antibodies. And these B cell receptors and antibodies are extremely diverse, and there's a number of mechanisms to support this diversity. The VD and J rearrangements for both the heavy chain and both the light chains, the combination of the heavy and the light pairings, and then the junctional variability, insertions and deletions that occur when these gene segments are joined together during development. And so already we have an incredible diversity that's greater than the number of B cells in the body for the potential repertoire. So after activation and somatic hypermutation, the orders of magnitude of the potential repertoire is vastly increased. So shown here are two examples of repertoires. On the left is a diverse one where each cell, each dot is a B cell, and on the right is an oligoclonal repertoire, that which you might see after an immune response, like 7 days after a vaccine. So we can ask questions about characterizing the diversity of these repertoires. We can look at particular lineages, which are groups of cells that are clonally related, like in the circle here. We could ask questions about comparisons between repertoires over time and ask whether there's clonal relatedness between sequences. So let me quickly describe some of the tools that we use to describe this from the beginning. I'm not going to go through this in detail, but this is our upstream pipeline. This is the work of Chris Pucilli and my group, where we take sequencing data, we have FASTQ files, and we process them through a variety of steps, including determining the isotype of these antibodies. So that's whether they're IgM, IgA, IgG. And we can do this for multiple samples. So we have sample A over here and sample B over here in this example. And this might be something like memory cells and plasma cells. We align each sequence to germline to determine which V and J segments are used and the boundaries between these joins. And then the critical part here is then taking this annotated sequence data and then combining them to estimate or have an approximation of how these sequences are clustered into lineages. And all of this data gets deposited into a SQLite database for further analysis. So the way the clustering works is fairly straightforward. We have multiple sequences, and we can shown here are 3 types of cell populations, and we treat them all together so that we can see whether there are clonal relationships between these populations. So we can put them initially into fairly broad buckets based on their V and J gene annotations and the length of their CDR3 regions. And this is an approximation of cells that have the same recombination event. We can then further subset these into clonal lineages by using pairwise sequence similarity for the CDR3 region. Now, the CDR3 region is that which is especially prone to somatic hypermutation. And then we can get these constructed lineages, which may include sequences from multiple cells or time points. And then we can have downstream tools that can analyze and visualize these data in many different ways. And I'm not going to go through these in detail, just show that we can look at the distribution of isotypes in our samples. So these are different cell populations and isotype distributions. We have different ways of looking at diversity. We can display the lineages from smallest to largest to determine whether these are clonal responses, like in the antigen-specific memory cells or plasmablasts, or diverse, like in naive B cells. And we can plot these using the Hill diversity functions here. So the naive cells are fairly flat, and the clonal responses drop up very quickly as you increase the order of the Hill diversity index. We could look at mutation rates. We can look at distribution of V-J combinations. We have J are the rows, V are the columns, and we can see there are non-uniform distributions of gene combinations. We could look at relationships between different cell populations. So here we have these clonality plots as the arcs, and we can look at ribbons between populations to show clonal relatedness. Or we could combine different attributes like clonality here, and we can use barycentric coordinates to indicate the different isotypes that are present, or distribution of isotypes within each lineage. And then these tools look a lot like R functions, that these are all command line. We supply a function and required arguments like a data source, or in this case, populations of interest. And then we can generate a figure instantly showing in this example, the relationship between memory cells and the same year and subsequent year antibody secreting cells for flu vaccine. We can write custom queries that allow you to ask very specific questions about your repertoire data. So I'm going to show 3 real quick examples of how these were used in publications. In the first one, this is part of a lupus study where we demonstrated that there's an alternative pathway for autoreactive B cells and lupus flares. And so normally it was thought that you would have the plasma cells that would occur during lupus flares could be derived from memory B cells, but we used repertoire data as well, along with other kinds of data to illustrate that there was an alternative pathway from recently activated naive cells. And that can be seen here, looking at over a time course. These are the distribution of clones and of activated naive cells, and the gold bars indicate clonal connectivity to antibody secreting cells. And especially during a flare, we see an increased amount of connectivity. And on the right, we're just showing for antibody secreting cells the ability to track these autoreactive or potentially autoreactive lineages over time. In another study with James Kobe here in infectious disease, we were looking at neuraminidase reactive antibodies after influenza vaccine. And this is neuraminidase is a relatively conserved component of the flu. And so there's a lot of interest in developing vaccines that might be more broadly activated or broadly responsive to this. And so in this example, what we're showing is the fact that when we include reactive monoclonal antibodies for neuraminidase in the sequencing analysis, we can see that over time, we have both day 7 and 1 year sequences in the same lineage. So it's evidence that, and then we can see this with CD138 positive, which are putative long-lived plasma cells. So this is an indication that these neuraminidase binding antibodies are long-lived and persistent in the bone marrow plasma cells. In another example, we have, this is the HIV human vaccine trial network. We're showing with 4 different vaccine strategies, we could compare using repertoire to compare the response, these different approaches. And in this example, we see T2 and T3 and T4 approaches have an enrichment of the VH1/2 clonotype or V gene, clonotypes with that V gene, whereas the T1 doesn't. And in other assays, it was shown that we don't get a lot of envelope protein reactive antibodies with this strategy. And so this is all consistent with the fact that these antibodies are largely 1-2. And this is shown on the right also that when we look at the lineages, each bar is an individual. And these are the different vaccine strategies. We clearly have an enrichment of the 1-2 lineages, especially for the 2 and 3 strategies. And like the previous flu study, we could look at monoclonal antibodies and see them in long-lived plasma cells several months later. So in the last 7 minutes or so, I'm going to switch gears and talk about a very different kind of process that we're working on. This is a cytometric bead array, which is designed to interrogate the seroreactivity to many proteins in human samples. So this is the work of Rodney King and John Killian in microbiology. They've developed this assay, and we're currently applying it to COVID. So we have many different beads. These are colorimetric beads that can be sorted by flow cytometry, and they come in different sizes. We apply, and each of these beads is linked to a particular antigen. And then we apply the serum sample, and then antibodies present in that serum sample will bind to the beads that have the antigens that are reactive in that serum. And then we can apply secondary antibodies, run it through a flow cytometer, generate a raw data file, and then ultimately we want to get a list of reactivities for the different isotypes and different proteins that we're looking at. And so what I'm going to describe is what happens in this purple arrow. So essentially what we're doing is using flow cytometry to first using automatic peak detection and gating is to identify the two different bead sizes using forward scatter and side scatter. And then within each of these buckets that are annotated as being a 4-micron bead or a 5-micron bead, we can then further segment the beads into different peaks. And these peaks represent the colorimetric beads listed here. And so we do this for each bead size. So this way we can increase the number of features we have in our assay. And then each peak will identify those beads that have a particular antigen linked to them. And then we can use the secondary antibodies to look at the mean fluorescence intensity and then get reactivity values. Sorry, my mouse keeps advancing here. So each peak corresponds to an IgM, IgA, and IgG response. And then for a given isotype here, like IgA, this panel of reactivities would appear here in this heat map where we're looking at a lot of longitudinal data from a single individual. And then the output of this pipeline is just a simple text file, or it could go into a database, which I'll describe in a minute. And it contains all the parameters used in the analysis, but here you have all your features, and here are all your reactivity values. And we compared this to doing this analysis manually using FlowJo, which is a fairly common flow cytometry analysis program. And we get high agreement between the two methods, between the quantification by FlowJo on the x-axis and automatic quantification on the y-axis. So this is now implemented as a high throughput process where you can queue up many FCS files. So typically we'll run several plates worth of samples in a day, generates one raw data file per sample. We can queue these up. We have a manifest file which describes metadata about the samples and indicates which of these samples are standard controls that we can use to make calibration curves. Then we also have other configuration files to define the flow assay here and the bead assay. So this is the linkage between bead peaks and antigens. And we also have a file configuration for the peak finding. You put all of this into this program and you generate all these files per raw data file. The ones that are standard curves are automatically picked out, assembled into a program which will then fit the data to a 4-parameter logistic curve to generate, so we can back calculate the concentrations, and then these data are dumped into a database along with their non-back calculated mean fluorescence intensities. And the key here is that this is extremely high throughput. So in our first run, we ran over 2,000 files, and going from raw data to complete compiled data set that we could start interrogating took under 7 minutes. And we're currently developing a database to manage all of these data. And the main way that we'll interact with this will be MATLAB, or we could build our tools to do this. And we're going to integrate this with the electronic health record extracts so that we can correlate the reactivity data with clinical parameters. So shown here are a couple of examples of how we can visualize this data. We can look at simple longitudinal plots for a single feature. So in this case, we've got days after admission for coronavirus on the x-axis. Then we have the concentration of the spike protein for IgA on the y-axis, and we can see a steady decline over time. We could also use a bar chart to show the profile, the reactivity to all antigens in the sample at a particular time point, and this is represented as a bar graph, and we do this for each feature and each isotype. Or we can combine these methods and show this as a heat map where the columns are days post-admission, and the rows are the features for IgM, IgA, and IgG. And so we can see these really distinct patterns for each antigen in the series here. So as you can imagine, there are a lot of queries we can ask. One of the first things we can do is take this really big data set and start with unsupervised approaches to see if there are ways that just using the data we can segment this and then see if these are related to clinical parameters, or we could use more direct supervised approaches to answer these questions with particular clinical features. So in summary, my group develops a lot of tools, mostly used in immunology, and we work with many different kinds of assays, both sequencing assays and flow cytometry or other kinds of arrays. And we're doing a lot, increasing amount of work in 10x at the moment. And we're always interested in exploring future collaborations with other investigators to apply these tools to new kinds of problems. And one thing I'd like to point out is that you can use, by having many of these collaborations, this is a great way to get requirements for future tool development and to make these more generalizable. And I'd like to thank many of my collaborators, my team, Chris Bucilli, Darrell Hill, and Mahika Kadem. And also the ones in yellow are those that I collaborate with on the projects that I talked about today. And I would like to thank them for this work. So thank you.
Thank you, Alex. We are running short of time, but I'll allow 1 or 2 questions. If you have a pressing question, you can ask. If no, if you have any other question, you can actually write your question in the chat box. So I'll move along. Our next speaker is Jelai Wang. He's an informatics architect at Informatics Institute. He's basically our go-to guy for any question regarding U-BRITE and the infrastructure. He's also a fantastic Python programmer. So he'll be talking about our U-BRITE infrastructure. And the title of the talk is U-BRITE, a biocomputing infrastructure to accelerate data-intensive translational science. Jelai?
Thank you, Malay. Appreciate the nice introduction. So good morning, everyone. Hope you're having a good ATTIS so far. So I'm definitely pleased this morning to get a chance to talk to you a little bit about U-BRITE, which is what we call our project to develop IT infrastructure in support of people doing data-intensive translational research. In particular for today, I'm just going to talk about what U-BRITE is and maybe show some examples of how it might help teams carry out team science. So I've just got a couple slides here. So U-BRITE is an acronym, and it actually stands for UAB, Biomedical Research Information Technology and Enhancement, which is a bit of a mouthful, so we all just call it U-BRITE. And it's definitely infrastructure about at least these 3 main things bulleted here. So one thing we're definitely trying to do is assemble clinical and bioinformatics tools that people think are important and make sure they're installed and work well on UAB's HPC infrastructure. So U-BRITE's definitely about getting the right tools in front of researchers in the right place. It's also about trying to help researchers with their data management and analysis pain points. So one of the big things we're trying to do with U-BRITE is experiment with and implement a design pattern called Commons. If you're not, excuse me, familiar with that design pattern, I did put a quick definition from one of the first papers to describe this design, and that's Robert Grossman's paper from 2016. And it defines a commons as an infrastructure that co-locates data storage and compute with common analysis tools. So it's a seemingly simple idea that I think we can all get a lot of mileage out of. So so you'll often hear us refer to U-BRITE as a translational research commons environment. And finally, we try to use U-BRITE to help facilitate team science, especially to the extent that technology can help people work together, excuse me, better as a team. So if you look at the overall architecture of U-BRITE, there's 3 main abstractions that we have implemented. So there's the idea of a data portal, there's an analysis gateway, and there's an expo gallery. And if you think about maybe a prospective new user coming to U-BRITE, how they might engage it, one of the places they might typically start is the data portal. A lot of users and teams might come to us with problems like siloed data or siloed code, or just generally, ideas that they want to share with their team, get feedback, iterate on faster, but they're just being blocked for various reasons, right? So one of the things we try to do is encourage people to start to engage the data portal, right? So they might begin by putting their source code instead of maybe at their desktop or notebook computer somewhere, to actually put it in a source code repo and immediately start using the well-documented and well-understood workflows within the Git ecosystem to start sharing code both within their team and with other people. Similarly, if they've got data siloed off somewhere, to start to put it in the omics data repo, or maybe if you don't have your own data, you can query our many locally cached public data that we try to put there. And so that's, just gives you some ideas about the data portal. But once folks do that, then that implements kind of the first part of the commons, right? So you've put your data side by side with the biggest compute and storage here at UAB, which is the Chiha supercomputer that's depicted on the top right. And then you can start thinking about some of the stuff I personally find pretty cool, which is, well, what's the right analysis environment? What's the right abstraction? And so if folks are interested in a web notebook style UI, they might look at Jupyter Notebook. If they're interested in doing more of an interactive research tool, then they might look at Streamlit or RShiny. But regardless of what a team ends up choosing, what we try to do with U-BRITE is, is assemble these tools, right? And so that includes tool building frameworks like Streamlit and Jupyter, but also just libraries of data science and AI and machine learning and visualization libraries, and try to make sure they run well within the commons. So before I leave this slide, right? So U-BRITE is about how this commons idea can help people achieve that fast R&D process that I think we all want. And part of how we do that, of course, is trying to remove obvious traps like those code and data silos, and maybe some of the obvious ceilings to scalability that we see teams run into, and try to provide a path to something that will avoid those. So for the rest of my 10 minutes, I'm actually going to do just a live demo. So we've got a what we call a U-BRITE technology demo, and that's located at demo.ubrite.org. And it's a live running web application that folks are welcome to come check out and play around with it. I wanted to show it today because it's a little bit easier to show some of this than it is to talk about it, especially some of the possibilities within the U-BRITE Commons. And so if you scroll down to the overview diagram in the demo, we try to highlight a few of the key interactions that we often observe for teams trying to do team science. And so you see along the bottom, there's the idea of data scientists or analysts and their role. There's the investigators, and then there's methodology researchers and maybe other collaborators of the team. And so what we're actually doing right here with the demo is we're pretending to be that middle category. So we're investigators interacting with this live running web app. Obviously, before we can do that, somebody had to write the web app. And so when I wrote the demo, I actually put on my pretend to be data scientist hat for a little bit. And so that's depicted on the left-hand side of this diagram. And I tried to author some Python source code. I used a tool building framework from the machine learning community called Streamlit. And I worked with the rest of the team, rest of my team within, in source code, within the source code repo to iterate, to get at something we felt like was representative of a real interactive data analysis tool. And so once we were happy with that, we deployed it to app hosting. And that's kind of what you're seeing here, right? A live running web application. And what you'll be able to see in this demo is that from this interactive tool, you can programmatically query out clinical data from I2B2. You can pull in analysis results. In this example, we're doing pulling in results from a gene expression analysis. And then we're taking the list of differentially expressed genes and we're passing it to a gene set method called Pager, which is a method from Dr. Jake Chen's lab here at UAB. So hopefully that's good enough for an overview. And I'm not going to go into a ton of details in each of these sections, but for the first part, we just show that we can query data out of the UWS API. And we show it here just simply as a table. But I think the more interesting thing from a data science point of view or scientist point of view is that it's a data frame and you can integrate it and combine it in different ways within the tool, right? So then the next section, there was a differential gene expression analysis that was performed by another part of the team. And so we wanted to pull in the results of that analysis and basically take that list of genes that were identified there and pass it to Pager, which is then going to tell us what gene sets or networks or pathways that might be of interest to us. So I think this is where the demo gets a little more interesting in that when we first visited this page, what happens, what had already happened was that we did all of those things. We made all those queries and we actually already passed those results to Pager, and it's already given us back 22 gene sets that we might be interested in. And this is with default Pager parameters. But if we wanted to tune it, so if we wanted to say we're only interested in gene sets of size 85 or larger, and less than 258, right? And then you can see it dynamically requeries Pager and gives us 12 results back. And just to show for completeness sake, we could also adjust some other Pager parameters, not all of them, just 2 representative ones. So these are just if you wanted to add or delete data sources, or if you wanted to adjust the false discovery rate cutoff. And so, so once as an investigator, you're happy with this, you might want to download it and play around with it further in another tool. So again, this is a technology demo, right? So it's not meant to be a real data analysis, but hopefully it's representative of what a team might actually do and want to do with an interactive research tool. So we're trying to set up the infrastructure with U-BRITE and put out examples to show people what might be possible in infrastructure to help with their team science.
Jelai, 2 minutes.
2 minutes, okay. So yeah, so for today, I just wanted to show you these 2 things, just to give an intro to U-BRITE and invite you to play around with this demo. If you're interested, it's at demo.ubrite.org. And if you want to find out more about U-BRITE, we've got a website at ubrite.org, which you're welcome to check out. We've got a lot of learning resources, training. We try to integrate with bioinformatics courses, various info courses, and others here at UAB to help people who want to use U-BRITE get started. So please don't hesitate to get in touch if you have any questions.
All right, so if you have any question for Jelai, please write it in the chat box. We'll move along. We are glad to have Greg Cooper with us. He's an investigator in HudsonAlpha. Greg's main interest is evolution and how evolution is affected in disease state. Many of you know that Greg is one of the person who is instrumental in developing a particular score called CAD score that we use regularly in analysis of genomes. Today, he'll be talking about long-read genome sequencing for diagnosis of neurodevelopmental disorders. Many of us are interested in long-read sequence analysis, so this talk is particularly pertinent for us. Gabe, welcome, and take it away.
Okay, great. Can I share my screen?
Yes, you can.
All right, can you all see my slides?
Yes.
Great. Okay. Thanks. It's fun to be here, and yeah. So as you heard, I'm talking about long-read genome sequencing, which is something that we're really excited about. And to set the stage here, one of the driving questions or goals in my group is understanding the genetic basis for neurodevelopmental disorders. And as a big picture umbrella here, we're talking it's something like 1 to 3% of children are born with one or more of the symptoms listed here. So intellectual and physical disabilities and seizures, autism, and other kinds of related difficulties that can pose some pretty significant challenges to both the child and their families. And identifying the root genetic causes of these conditions, which are very often individually rare Mendelian disorders, is really crucial to best counsel and treat the affected children, so to come up with the best therapies we can to help those children, but also to counsel their families about what to expect and how to best manage their child's symptoms. And then, of course, in the long run, genetics is powerful at targeting the underlying root causes in terms of identifying new drugs, new therapies, new strategies to improve outcomes down the road in the long term. So at HudsonAlpha, we've been very interested for many years now. So we've just showing you a list. We started really in 2013 with Martina Bevan, who's a really wonderful UAB pediatric neurologist who spends a lot of her time at a clinic in Huntsville. But we work with other partners, including CRS, which is an Alabama state program. Right now, we have a grant called SouthSeq, which is looking at newborns that are in a neonatal intensive ICU with symptoms of a congenital disease at 5 different NICUs across the Southeast. We're part of what's called the Alabama Genomic Health Initiative, which I'm sure a lot of folks have heard of, or AGHI. This is a state-funded collaboration between UAB and HudsonAlpha. And the basic sort of script here, the process that we generally use is that we identify children with an undiagnosed condition, undiagnosed disorder. We subject them to Illumina genome sequencing. This is sort of the standard collection of short reads to get to roughly 30x average coverage. And then we use to assess individual variants in terms of whether they're relevant to the child's symptoms. We use the classification rules and system devised by the American College of Medical Genetics, or ACMG, that allows us to assign evidence codes and basically have structured vocabulary for how we classify variants. And to summarize a whole bunch of work that we've done as part of these collaborations, we've sequenced close to 1,400. This number's a little bit out of date. It's a little over 1,400 now. Probands, we've sequenced a total of 2,800 people because we often sequence mom and dad as well, because their information can be valuable at understanding the child's, once you understand inheritance, that can help you interpret a variant. So we often, but not always, sequence trios. And this pie chart here sort of summarizes the net results of that. And so we found likely pathogenic variants. So this is PLP for short. These are ACMG designated terms. And essentially, these are variants that are, we believe, are definitive or highly likely to be causal for the child's symptoms that prompted the sequencing. And so these are effectively the category of variants that lead to allowing the clinician to make a diagnosis that it can be used for management, treatment, and other kinds of clinical decisions. So you can see a little over a quarter of children. These are, as I mentioned, these are individually rare. So as we do see some conditions pop up repeatedly. So here's 2 examples of SCN1A and MECP2 associated with Dravet and Rett syndromes, respectively. We've seen each of these a handful of times, but we've seen over 220 different genes. So among these 370 children, the majority of them, they're for their particular condition, their particular diagnosis, that's the only time we've seen that over the course of all the sequencing that we've done, just showing it that, again, these are all individually rare conditions. Most of these are de novo, but we certainly see other kinds of inheritance, recessive, dominant, transmitted, and also X-linked. And I say there's also a bunch of VUSs, and I won't talk about that today, but this is a substantial fraction of probands end up with these. And these can be frustrating, obviously for the families, because they're intrinsically uncertain. We're telling them we found this thing, we don't know what it means, but we think it could be relevant. But they're very, they're high priority targets in terms of research, because these are mostly mutations in genes that look like it would be bad symptomatically if you had a mutation in that gene, but we just don't know enough about it. So a lot of our research is on characterizing those VUSs. But today I really want to talk about the fact that despite the fact that we keep getting better at this, we're still failing more often than we succeed, right? So this is a majority of the children that we cannot find any variant, even an uncertain one that could be relevant. And right, so this is no plausible causal variants found in many of these kids. In fact, the majority. This is similar to literature from other kinds of, lots of other studies in both pediatric and adult conditions that look Mendelian, that exome or genome sequencing just cannot find anything. And so the bottom line is that we believe that there are likely to exist a lot of highly penetrant mutation events that we just aren't seeing right now. And so that leads to this prediction that if we can do a better job at more comprehensively discovering variants, that could unlock many rare disease discoveries. And so we started a partnership with Jane Grimwood and Jeremy Schmutz at HudsonAlpha. They are fellow faculty members there. And they, for a long time, have been the directors of the HudsonAlpha Genome Sequencing Center. This is a Department of Energy funded group. And primarily, especially over the last 15 years, their focus has been on assemblies of plant genomes. So if you're familiar, if it's a agriculturally or bioenergy important plant, odds are Jane and Jeremy's group is the one that generated the data that led to the reference assembly of that. So cotton and soybeans and poplar trees and all kinds of things. And because of their interest in assembly of complex genomes, they have expertise in infrastructure for generating assemblies, which includes both short and long reads, and also a lot of the intellectual expertise and computational work power to generate assemblies. And as part of that, they were beta testers for something called circular consensus sequencing, or CCS, from PacBio. This is the brand name is called HiFi. And what's neat about PacBio CCS is, if for those who are familiar with long reads, historically the major downside to long reads, obviously the upside is that they're long, right? If you go to thousands or tens of thousands or even hundreds of thousands of bases read length, that can give you visibility into the genome that 150 base pair reads just can't. But that came with a cost of having very high error rates. So any individual base call had something like a 20% chance that it could be wrong. So the way CCS gets around this challenge is that they start with these long high molecular weight fragments. They attach these adapters to them, which basically circularize the molecules. And then instead of sequencing one long molecule one time, they have this polymerase loop over that circle over and over and over again. And you can see here what you get is this looping where you're reading the same positions repeatedly. And so basically what this allows you to do is you can identify a consensus base at each position from collapsing the repeated observations at the same locus. And basically the errors, to the extent that they're random, not all of them are, but many of the errors are just random, that washes out if I read a base 2 times, 3 times, 5 times, 10 times, et cetera. And so what this allows to happen is that you get reads that are both long. So we're talking about tens of kilobases long, but now they're accurate. In fact, they're as accurate as Illumina reads are at the base pair level. So these have less than a 1% error rate in terms of per base pair call. So what we applied was we generated CCS data for 6 probands that we had previously studied using Illumina genome sequencing, in which we could not find anything. So you can see here the symptoms here. Each row here is a proband that we studied. Pretty typical representation of the phenotypes that we're interested in: intellectual disability, seizures, et cetera. All of them had previous genetic testing, exomes, genomes, or arrays. And I'm not showing you everything. A lot of these kids have undergone this, a diagnostic odyssey with lots and lots of tests. But you see we generated fairly high coverage from CCS reads, so from a mean of 25 to a 35x coverage, and we have intermediate coverage of parents. So we shot for about 15x coverage for mom and dad. with the primary goal being to use their data to assign inheritance of variants that we found of interest in the proband. So we took a couple different analytical approaches here. So obviously you can treat them like Illumina reads. You can align the reads to the human reference, in our case HG38 with BWA mem. We actually perform variant calling with Google's deep variant algorithm, structure variants with PVMM and PVSV. These are PacBio software packages. What we found was exceptionally high quality calls of normal variants. So if you just look at, for example, de novo point mutations or indels, on average, each human baby is born with something like 70 to 100 de novo point mutations and indels. And so you can see we're seeing about 89, which is right in the range of biological expectation. In contrast, I'm not showing the data, but from Illumina data, you typically get several hundred. So you get lots of erroneous de novo calls. So we're already seeing evidence that we're getting higher quality variant calls. But one power that you just simply cannot do at all with short reads, or at least not at all well, is that we can go and generate assemblies on a per proband basis. So we use this approach called Hi-Fi ASM. And what's neat about this is that this is done in a trio aware fashion, so that you can take contigs that are generated for the proband and you can phase them and assign them to maternal and paternal haplotypes by using the sequence information that you have available from mom and dad. So here is just an overview of the assemblies that came out of Hi-Fi ASM. It's really remarkable. So for each of the 6 probands here, I'm showing you the contig N50. So this is the length of contig at which half of the assembled bases are included. So you can see we're getting N50s that are over, that are 15 megabases-ish to up to 40 megabases or so, and they're parentally aphased. So we can separate mom and dad's chromosomes here, and you can see our N50s are essentially chromosome arm lengths. It's really remarkable degrees of contiguity that are coming out of these assemblies. So I just want to focus on some neat examples here. And obviously, the assembly is great, but our primary goal here was to find diagnostic variants. So one of the first things we noticed is this region of the genome that includes CDKL5 exon 3. So here is, along the x-axis here are HG38 genome coordinates, and we're showing you IGV. So these are alignments of the CCS reads for mom, dad, and proband to the reference along the x-axis. So each read here is a row. So where it's gray, those are stretches of those reads that match to the reference. I see mom and dad, I'm not showing you all their reads, but mom and dad effectively appear to be identical to reference. But you can see the proband has these subsets of reads where you have this rainbow pattern here, where these are stretches of reads that look normal for long stretches and then transition to some different sequence. And so this suggests to us that there's something happening. A mutation has occurred near this, affecting the CDKL5 exon 3. And so, of course, that is hard to understand what's happening here. One way that this gets resolved very quickly is if you look at the assembly. So one way we do this is the so-called dot plots, where we're looking at stretches of similarity between, this is the proband's contig here on the y-axis compared to HG38 on the x-axis. And often what you see is these diagonals. These are long stretches of sequence where the proband's assembly is effectively collinear and similar to the reference assembly. But you can see here we have this gap where there's a chunk of sequence present in the reference, present in the proband's content in the reference. And you can also see that it appears at least partially similar to this downstream region of the reference assembly. So when you go in and annotate this in detail, what you get is this. So the proband has an insertion of about 7 kb. It's partially an L1. This is a mobile element. So this is basically our genomes are filled with mobile elements like ALUs and L1s that are still active in human populations. So this is a brand new mutation event that resulted from an L1. But it also actually transposed a downstream segment of intronic DNA. That was that duplicated chunk I showed you of PPEF1. So it's a complex insertion event composed of multiple pieces. The important part in terms of disease status is you notice that CDKL5 exon 3 is here. And it actually, the insertion event creates a duplication. So this is called a target site duplication, where a chunk of sequence near where the insertion inserts is duplicated. And in this case, it includes the entirety of exon 3. And so you can see this is amplified RNA. where the proband has this, it's faint here, but you can see it as a band that's shifted up relative to the normal band that you see in proband mom and dad here. And when you cut that out of the sequence, that indeed includes an extra 35 nucleotides. And the net result is that that duplicate exon, because of 35, leads to a frame shift, leads to loss of function, which is in this case, CDKL5 loss of function mutations are a well-established cause of developmental and epileptic encephalopathy. So this is considered likely pathogenic and essentially becomes diagnostic for this proband. So I want to go quickly through a second proband, for the sake of time, I'll skip a lot of the details, but here I'm showing you, again, dot plots where we're looking at chromosome 7, a chunk of chromosome 7 from the proband and a chunk of chromosome 9 from the proband compared to 7 and 9 from the reference. Long story short, proband 7 includes derived chromosomes, which are actually essentially translocated or rearranged pieces of 7 and 9 translocated together, along with a little chunk of an inversion here. You can see the dot plot is flipped on its axis here, so it's an inverted sequence. And when you again resolve all the mechanics in this is what the child's maternal chromosomes appear normal. So these two, 7 and 9 here in pink, but then the derived 7 and derived 9 inherited from dad are significantly rearranged here, where you have a chunk of chromosome 9 inserted into 7, and then you have the reciprocal event on the chromosome 9 along with an inversion event. On top of this, there's a lot going on in this child's chromosomal picture. We saw extensive rearrangement of chromosome 6. So you can see right away, this is a dot plot of the child's chromosome 6 against HD38. We have this massive inversion here. And we also have this region where if you zoom in, you'll see a whole bunch going on here. So we have inversions, we have shuffled events. And so basically this looks like chromothripsis. So to summarize that, we have this large pericentric inversion with a 10 megabase chunk where we have this, basically it's like the genome was shattered in this region and then put back together in the wrong order and orientation. So it's a really extensive amount of rearrangements. And so again, this structure variation found in this child was also considered likely pathogenic. And I should say that this is glossing over a whole bunch of work. It was really a heroic effort, especially from Susan Hyatt and James Lawler in the lab to sort through these assemblies and figure out how all these pieces fit together. So with that, I'll just conclude that we've generated long-read genomes from 6 trios. The assemblies were fantastic and were phased and just wonderful data. I didn't talk about this, but it really is remarkable how accurate we can detect variants, especially in complex regions like segmental duplications, mobile element insertions, et cetera. 2 out of 6 of these have harbored a diagnostically relevant variant, which again, small number, so we don't know that the rate is 1/3. But certainly it's consistent with the idea that a substantial fraction of currently unsolved probands harbor diagnostically relevant variants that we might pick up with long reads. And so we're really excited about where this goes. And with that, I'll conclude and just say thanks to all the families that participated. Obviously, we can't do this without their generosity in participating in research. And Susan and James led a lot of this work, but then my whole lab and our collaborators are all important. And yeah, that's it. There's quite time for questions. I take it, but I can move on.
Thank you, Greg. If you have any question for Greg, you can ask.
Wonderful talk, Greg. A quick question. How was the samples were selected for long-read sequencing? What criteria were used for this for selecting these samples?
Right. So yeah, the short version is that they had to be they were samples that we had in the freezer that were negative by all of our previous attempts, but that basically we use our clinical collaborators to give us their best estimate, their best selection as to which children they think when they look at their phenotype, and these are all kids that they've treated directly, so just their sort of intuition that is likely genetic. So it's not a random sample by any stretch. It really is. We wanted to maximize our chances of getting a hit, so we asked them go through and pick out the samples that your hunch is that it's genetic. It's not really an objective score criteria. It really is an intuition that they gave us. My sense is that obviously they were picked for a non-random reason. My sense is that there's a whole bunch of positive examples that we're going to find in these negative cases, but certainly if that was part of the criteria is the clinician's intuition.
How many?
Yeah, go ahead, Jen.
How many unsolved cases can you envision could be solved by long-read sequencing? There's over 70%, right? But I think not all were due to structural variations or these complex rearrangements, right?
Right. So yeah. So the bottom line is right now it's let's say that all the VUSs that we know of were to turn into pathogenic, which is not true. The maximum diagnostic rate that we currently see from Illumina is on the order of 40%. So it's that 60% is that. So what fraction of that 60 will resolve with long reads? I have no idea. Right now, I would say 2 out of 6. Right, one third is my best guesstimate. Obviously, those are still too small accounts. It could be quite a bit higher or lower than that. But certainly, my guess is that it's a substantial fraction. I certainly firmly believe it's likely not to be a majority of the unsolved cases, but I would say a substantial minority.
I have a question, Greg, for you. This is slightly off topic. So many of these cases where you do not find anything, like in this case, for example, you find that there is this rearrangement event that was not detectable. But what is your opinion about having synonymous mutation having an issue in any of these cases? Like, for example, nowadays, there are work going on, particularly in cancer, the role of translational selection and synonymous mutations. So do you think any of these disease synonymous mutations can actually have a cause that you cannot detect?
Absolutely. So I think there's still a lot of mutations, a lot of diagnostic information that is available, that is inside the Illumina, the standard variant calls that we just aren't capable of interpreting yet. So synonymous variants, regulatory mutations that affect non-coding sites. There's this whole class of element that we've been working on recently called poison exons, or this weird molecular mechanism that isn't really well understood. So I have no doubt that there are variants that have a strong molecular effect that lead to disease that we just, because we don't have a sufficient knowledge about how those elements or variants work, we can't interpret them correctly. So I'm certain those exist. What fraction of unsolved disease are they? I really don't know, but certainly that is a good bet for at least some of the cases. Absolutely.
Any other question for Greg? If not, I think Greg will stick around, right?
Yep, absolutely.
Yeah, so you can ask Greg, and we can actually, in the break, we can actually have a little bit of discussion if you're interested. So we'll move along. Our last speaker for this session is Maggi Chen. She's a PhD student in Informatics Institute, and today she'll be talking about Inspector, a reference-free long-read de novo assembly evaluator. Maggi? Is Maggi here?
Yeah. Can you hear me?
Yes, go ahead.
Okay, cool. All right, so hello everyone, my name is Maggi Chen. I am a graduate student at Dr. Zheng's lab. And today I'm going to talk about my project, Inspector, accurate reference-free evaluator for the de novo assembly. So starting with the background, de novo assembly has been a very popular tool and method, and has been widely applied in many biological studies, including the variant detection just now. And a high quality assembly is the foundation for the accurate downstream analysis. If you have a lot of errors in the assembly, then all the results you get will be unreliable based on that assembly result. So it is very important for us to evaluate the assembly result to see how accurate it is, and also to know where are the assembly errors. Very few tools can be used to evaluate assembly result. We have Quest, which is a reference-based method. They compare the context to the reference, and they report the misassembly every time they see difference between those two. So they cannot really distinguish the genetic variants from the misassemblies, and the number they report will be much higher than the true number of assembly errors. We also have the Mercury, which is a chemo-based approach. They don't require a reference genome. They also use the read to evaluate the assembly context. And instead, they report the location for the problematic keywords instead of the exact location for the assembly errors. And also, they cannot deal with the repetitive regions that well, because all the keywords in those regions are occurring multiple times in the genome. So in order to fix all the problems and evaluate the context accurately, here we propose Inspector. Instead of relying on the reference genome, Inspector used the sequencing read to evaluate the context and also to identify the assembly errors. So there are 3 major steps for Inspector's evaluation. We have the read alignment, and then error correction, error identification, and eventually an optional error correction step. So first, the reads are aligned to the context with minmap2, and then we can calculate a matrix for continuity analysis, including the number of contexts and 50 mapping rate and other stuff. And then we will identify the structural errors that are longer than 50 base pairs, and also the small scale errors which are shorter than 10 base pairs. Both the structural errors and the smaller errors will be distinguished from the genetic variants based on the ratio of error supporting read. So a true assembly error will have about 100% of the read supporting this event because both haplotypes are different from the context. But if we are looking at genetic variants, only one haplotype is different. Then only 50% of the reads from that haplotype will support error event. And by separating the 50% and the 100%, we are able to remove the genetic variants and only keep the assembly errors here. So that's how we identify the errors. And since we already know the location, size, and everything about the errors, we are able to really correct both the structural and the smaller scale errors, and then report the corrected context with higher overall accuracy. So that's the workflow for inspector. And we first did the benchmark in the simulation data set. We simulated a human genome with the SVs and SNVs, and we simulated the assembly context based on that genome with the assembly errors like the structure and smaller errors embedded in that. We also considered the haploid and diploid genome and also simulated the hi-fi or the CRR reads for the inspector. So we applied the 3 different assemblers and evaluators and see if they can identify those simulated errors accurately. So here, if we look at that F1 score, which is the error detection accuracy, we see the inspector is having consistently high accuracy throughout these 4 data sets for structural errors and also for the small scale errors. And especially for the CRR data, which has a lot more sequencing errors than the hi-fi data, we see a lower recall, about 86% than the hi-fi, which is 99%. But still, we have very high precision, and that leads to a high accuracy, over 90% for the small-scale errors. We also evaluated the simulated dataset with Mercury, and they were able to identify about 70% of the errors with a precision of 90%. It's not as high as Inspector, but it's pretty good. And we also have Quest. Quest is reporting a lot of SVs in their misassembly set because they are reference-based approach, so they have lower recall and precision than the other 2 evaluators. So here we show the inspector is having very good performance in the simulation data set. So then we moved on to the real data. We assembled a human genome of H002 from the Genome in a Bottle project, and we used the CR and hi-fi and the nanopore data to perform the genome assembly. We tested 5 different assemblers, the Canoe, Fly, WTVG2, hi-fi assemble, and Shasta. And here we show the number of errors for both structural errors and also the base errors per million bases. So we also calculated the quality score here based on the number of errors we have identified. And this represents the assembly quality. A higher score indicates a better overall accuracy. So if we compare the performance of different assemblers, we see Canoe is having the best quality score for the CRR assembly and also for the hi-fi assembly. And in the nanopore assemblies, Fly instead, they get the best result. And they actually have a significantly fewer number of errors than the other 3 assemblers. And here in the H002 assemblies, we also evaluated each assembly with the Mercury. And here we list the quality score from Mercury's result. We compare the quality score by inspector and by Mercury, and we see they are highly correlated, even though they don't have the exactly same value because they are calculated by totally different algorithms. So since the inspector is able to report the exact location for the assembly error, we can perform repeat annotation for these errors. About 59% of the human genome is covered by the repetitive sequence. So we would expect to see about 59% of the errors located in the repetitive sequence if the errors are generated randomly. So that line is marked here. That is the baseline we are looking at. Here we use a bar plot to show the percentage of errors located in the repetitive regions for each assembly and for the structural errors and the small scale errors. And we observe the value to be always higher than the baselines throughout each assembly for both error types, which means there is enrichment of the assembly error in the repetitive regions. We performed the one sample t-test to check if the difference is significant, and we did get a very small p-value for both the structure and for the small scale errors, which means our inspector is able to identify errors even in the repetitive region, and also to show the enrichment of assembly errors in those regions. So that kind of suggests that is like the assemblers that are facing more difficulties in terms of assembling the repetitive sequence than the unique sequence outside of the repetitive regions. That's why there is a preference for the assembly errors. So eventually we have the error correction function. So we applied error correction with the hi-fi reads, and then we evaluated the original and also the corrected assemblies to see if we have improved the quality of the assembly. So here we have the number of small scale errors and also the number of structure errors. The gray is the original assembly and blue is the corrected. And we observed a very large decreasement in the total number of errors in both small and larger errors, especially for the smaller ones. So in the nanopore assemblies, we are able to correct about 90% of the total base errors. And for the CRR assembly, we corrected about 80%. And for the hi-fi, it's only 30 to 40%, as this one is already very accurate even before the correction. So there isn't too many errors to be corrected. So here we calculated the quality score with inspector to see if the corrected assemblies have higher overall accuracy than the original ones. And we did observe the higher score in the blue dots than the gray all the time, which means there is really an improvement here. And in the last figure, we cross-validated with mercury just to make sure that with the chemo-based assembly evaluation method, we are also validating those improvements in the overall assembly quality. And we did see that the blue dots are always above the gray ones. So here we are showing that by performing the error correction with hi-fi data, we are able to reduce the number of structural errors and also the small scale errors. And we did observe the increment for the quality score, either by inspector or by the mercury. So, little conclusion. We are presenting inspector as an evaluator without help from the reference, only relying from the sequencing read. And it can report the assembly errors very accurately, and also show the enrichment of the assembly errors in the repetitive sequence, especially in the simple repeats. And we also have the error correction module, which can improve the assembly quality and hopefully help the downstream analyze a little bit. All right, that's what I have today for inspector. Okay, thank you all.
Thank you, Maggie. We really do not have time anymore for questions. We'll have exactly, I think, 10 minutes or so. So we'll come back at 12:15 for the next session. And thank you very much for all the speakers. It was a great session.